네, 안녕하십니까?
Good morning, everyone. Welcome to the BIX 2022. We are now talking about how far and we in AI-based drug discovery. So we're going to have our regular session number 1. As the slogan of this conference is Summer in BIX, I really hope that you can beat the heat. Actually, today it is rainy a little bit in the morning, so it is a little bit better than yesterday in terms of the temperature. But despite the weather, which is rainy, thank you very much for coming here. So I'd like to now start our session in earnest. Recently, across the whole industry, we do have the research and development that are underway using AI, artificial intelligence, especially when it comes to the global AI-based drug development market. We have the 1.6 trillion won, which is 1.43 billion US dollars up until 2024 in terms of the growth. That will show the CAGR of 40.8%. Starting from the ingredients and IND, we have the time taken until these clinical trials. We can save the time with the AI, so we can have the session in order to know where we are in terms of the AI-based drug development. So we would like to show you how advanced or how less advanced we are. As of the first half of 2022, the business models based on the AI are represented by 500 companies in the market. Not all the AI-based new drug development companies have their own expertise, that they are the best in terms of that. What is important is who can be the best one to give the best solutions to the patients. How can we anticipate the regimen and the treatment plan for the patients, and who's going to be the first one at the racing? That will be the most important thing. So we have the honor of having the experts in this area, so we will listen to them about the market, and we will also ask questions if we have, in order to identify where we exactly are. In terms of the market, which is the most promising one, so we can find out what is the direction desired by the industry, and also along with the processes of having the drugs development, we can also listen to the reality of the BT industry. So I'd like to have now Mr. Woo Sang-wook from PharmCat to talk about the AI-based technology, please.
네, 반갑습니다.
Thank you very much. As introduced, I am Woo Sang-wook, CEO of PharmCat.
장소에서 멋진 분들과
I'm very honored to be before you and deliver my presentation. And I would like to thank the organizer, BioPlus, for giving me this opportunity. Let me begin my presentation. The title of my presentation is AI and Quantum Physics-Based Drug Discovery. So here you can see the content of my presentation. I will first talk about the current trends in AI-driven drug discovery. Then I will give you a very brief overview of our company. Then I will explain you about the platform technology that we have. And I'll be finishing my presentation in 20 minutes. This slide shows the 2020 Nature Review Drug Discovery. So this is a very latest trend. And this shows how many AI native firms, this is based on the data disclosed by AI native firms. And you can see that as of 2018 and 2019, actual discovery and preclinical materials and substances, you can see that there are hundreds of them. And the blue indicates the phase 1 and phase 2, and you can see some of them as well. So I think that 2019 has been the inflection point, and after that there was a massive increase in AI-based drug discovery. And also the table on the right side shows the target comparison between the large firms and small ventures, and you can see that the targets are actually overlapping. So the kinase and the non-kinase enzyme, as well as GPCR, are now leading the trend. So AI-based firms and the big pharmas, they do have the overlapping target areas. And also, if you look at the therapeutic areas, again, there are a lot of duplication overlapping, with the oncology topping the trend, followed by CNS, immunology. Especially when it comes to immuno-oncology, it's really emerging fast, and the trend is more toward oncology and immunology nowadays. So these 20 firms, I think they are really leading the trend. And you can see that the AI-based firms, they actually set the trend and lead the trend, even for the pharma industry. And now, so the PharmCAT is based on AI technology, but our basic technology is with the structure, so structure-based drug discovery. And from such company's perspective, the target protein and also understanding the 3D structure of target protein is very important. So external, MMR, and the cryonic, this is the way that we understand the protein structure, but they have not been crystallized yet, and therefore in order to design drug, we have to have a prediction based on AI. As you know, the AlphaFold 2, this has made a tremendous job in this regard. So more than 200 million proteins were predicted by AlphaFold 2. So as of 2020, you can see that after the AlphaFold won in Cas4 competition, there have been the massive increase in the number of research papers that cite the AlphaFold 2. And also the target proteins, you can see that there are a lot of AI-based researches. And Schrödinger, they also announced that they started the phase 1 clinical trial using AI-based solution. So this indicates that a lot of AI drug discoveries have come to the real-world scene. And so we established our company back in 2019. So we have about 3.5-year history, and we have 2 co-founders, I, myself, and also Mr. Kwan. And as we are in the year 3, we have done a lot of activities, but they are mainly related with technology, so I'm not going to delve into that. Let me briefly introduce our company to you. Our basic direction is AI and quantum physics-based platform. So we want to develop a platform for new drug discovery and development that are based both on AI and quantum physics, and our platform is called Formulator, and this is for small molecule medicine. This is something that we have done since our inception. And based on that, we actually expanded our platform to DDS and RNA with PharmBag. And also even for PROTEC, we have established a platform called PharmTech. And also we are now establishing the platform for ADME, which could be the basics for various businesses. And in order to develop these platforms, we have to have a lot of human resources as well as the computing power. So we have over 35 PhD level scientists in various fields. We have the quantum database and network database for proprietary database. And nowadays, the bio investment has reduced significantly. So in order to be a NCDO, our target is to focus on clinical phase. And after the outbreak of the COVID-19, together with the IZIN, we have developed the COVID-19 sequence based on mRNA, and we are now in phase 2 clinical trial for this. And among the 4 business areas, now I'd like to talk about the formulator, which is a platform for small molecule medicine development. So I'll briefly talk about our technology. Formulator, this is a AI and physics-based technology. And the physics here refers to not just the physics, but the chemistry and biology. So the formulator basically consists of the 5 platforms. The first 5 tools. The first tool is the protein 3D structure optimization and RNA structure prediction. And we start with the structure basis, and therefore, when selecting target is very important. When we do the collaboration, usually the partner firms give us the targets, but sometimes we select our own targets. And once the target is set, then X-ray structure and cryogenic and MNR structures, we see whether the target does have these. And if there isn't, then we just start, and if not, then we make prediction. So the AlphaFold, yes, that's excellent, but it could be different from the protein that you want to build. And therefore, we have developed our own protein prediction module. So if the AlphaFold is not sufficient, then we run our own prediction and simulation program. And once we have the 3D dimensional target, then we do the screening. So usually based on the Camry or the Zinc database. And about 100 servers that we have, these are dedicated for screening only. And we can screen about 12 million database. And once the small molecules are screened out, then what's important is how compatible it is with the protein that we want. That's why the docking is important, and a lot of pharma companies are using docking programs. However, docking programs do not really take full consideration the various living conditions. So, like in terms of the temperature changes, or the envelope. So that's why we use the virtual screening and MD simulation for massive decking. And molecular dynamic. So this is to look at the affinity between the target protein and the substances. And the quantum calculation is based on the Schrödinger function, so that the small molecules' interaction can be monitored at the electron level. And so for this, we have established the quantum database for about 3 years, and now we can have very fast and rapid calculation based on this technology. And once we have that, then for the substances that are screened out, we have to see whether they are toxic or not. So based on the existing database and using machine learning methods, we see the hepatic and also the renal toxicity. And if there is any red flag, then we just throw them away. And if there is no toxicity found, then we go on. So once we reach the stage of the ADME-T, then so we usually start from the 700 million, but by the time when we reach ADME, there are only 40 candidates. And then the bioassay team, they conduct the in vitro assay out of these with these 40 candidates. And then the substances that are selected in this phase will go to the drug generation phase. So with the AI-based automated process, we generate a lot of drugs, and then we run them on the formulator. So the new novel compound with a great binding affinity and no toxicity is something that we produce out of this formulator. And then we do the synthesis together with our partners. And so from target to drug, you can see that we do have this kind of clockwise forward process. And another unique feature of the formulator is that when we work with the research institutes or the pharmaceutical companies, sometimes we find that the target is not known, although the efficacy is great. Then in such a case, we do have the protein-protein interaction network and protein-drug interaction network. So using these systems, we actually identify the targets, expected targets for the drug substance. And
리버스 프로세스를
we also have the reverse process where we can create the products even better than what we initially designed. And this is for the targeting factor 9. So for the initial factor 9, here you can see the zinc, and we did the screening. So we did the screening of the 700 million candidates. And I don't know whether it's readable, but there is a Gaussian distribution, and this is a binding affinity that are produced from the screening process. And then among them, we select the best one, and then we do the AI generation. And then we find the best candidate with the best binding affinity. And you can see, so it has an excellent binding affinity, but it has very different forms of the compound. So here you can see that the AI actually helps us a lot in producing and developing a new compound. And this is the reverse process. So this is actually the example when we partnered with the Korea Chemistry Research Institute and Nuclear Research Institute. So we identified a target based on this reverse process. And we actually create the better substances with the AI technology, AI compound. So we generated the 10,000 AI-based compounds, and then eventually we were able to produce these 3 different substances, and they are currently under the preclinical trial, animal trial. And this is a PharmBag platform for sequence optimization and RNA encapsulation. So this is for the COVID-19. And our concept in developing and carrying out this project is that we want to create a platform for more efficient production of vaccines. So again, aligned with the same philosophy with the formulator, we produced this PharmBag platform. And it has 4 modules. The first one is the RNA structure prediction. So this is for the sequence optimization and RNA structure prediction. So this is a 2D or 3D prediction based on AI. So this is the first module that's about the RNA structure prediction. And then once your RNA is injected into the human body, then usually they are degraded because of the enzyme. So we create a drug delivery system that prevents that. And especially we use the LNP or the lipid nanoparticles. So we have a module for the drug delivery system optimization using lipid nanoparticles. And once we have that, then we think about the DDS and RNA encapsulation. So this is the third module. And then once they are injected into the living bodies, they should survive and avoid escaping. That constitutes the fourth module, which is RNA endosomal escape. And this is, and based on that, we do have the multi-scale MD simulation based on quantum calculation and atomistic coarse-grained simulation. So here, you see this is, here you can see a very complicated picture of the lipid nanoparticles. A lot of pharmaceutical companies, they are highly interested in LNP, especially ionizable lipids. And they are also interested in the patents surrounding them. And competition is very fierce in this field. And in order to secure the patent and create substance, we also use AI and simulation. And on the right-hand side, you can see the quantum, electronic level, and also coarse-grained multi-scale simulations. And this is just too detailed, but let me briefly mention that. So this is something that we recently launched after long years of research. So LNP, when it is fused with the endosome, then how it is evolved. So using simulation, we show this evolution process. So when it comes to the ionized LNP, depending on the double bond, the fusion efficacy or efficiency could be different. So let me show you this video. I don't know whether it will work. So as you can see through the coarse-grained system, LNP and endosome are fused.
이렇게 해서 퓨전이 들어가면서 두 멤브레인이 만나는 걸 볼 수 있습니다. 그죠? 그다음에 이제 그 오른쪽 것도 보시면
And also on the right hand side, again you can see a different thing. So sit and any, there is only one difference of double bond, but here you can see what actually happened is pretty different. So we research on this.
그래서 퓨전이 되면서
So when they are fused,
그래서 이런 것들을
you can see that it is completely get into.
이게 지난번 저희가 7월 14일
And this is the AI drug discovery conference that we held on July 13th. So we brought together experts around the world to hold this conference. So in Netherlands, from Netherlands, Canada, Italy, and France, we have invited the researchers and collaborators, and in order to hold this conference on drug discovery.
이제 저희가 또 이제 지금 굉장히 열성적으로 연구하고 있는
And another area that we are highly interested in is the PROTAC. As you know, the small molecules, they either inhibit, mostly inhibit target proteins, but in some cases, the inhibition itself could not be effective, and therefore, in these cases, elimination of the target protein could be more efficient. And in fact, in our human body, we do have the mechanism called proteonism that, or E3 ligation, that eliminate the target protein. So if you link these inhibitors and the target protein that you want to eliminate, then you can have this PROTAC. And PROTAC consists of three things. First of all, you have to have a target protein that need to be eliminated, and this is what we call warhead. And E3 ligation, and also the liquidum that is linked with the E3 ligation, and also the linkage. So these three things should be well designed in order to have the successful PROTAC. As I mentioned earlier, our company has developed a platform called Formulator, and this is for small molecule. So we actually had an extensive research on AI-based small molecule research, so which consists of the warhead part. And our unique differentiation from the PROTAC company is that usually the PROTAC players, they just research E3 ligations and the target proteins, but here you see, we research the entire full machinery process, entire comprehensive process, and we have created the model already. And based on this model, we are trying to have an effective ubiquination of the target protein that needs to be eliminated. And we also researched the linker as well. And recently, what we are interested in is that there are about 600 kinds of the E3 ligations, but the structures are pretty simple, only two. And because we are the AI-based solution provider, and therefore we want to recruit a new E3 ligase. And these are our pipeline. So we have, through the farm bag, we are developing the COVID-19 therapeutics, vaccines, and also the cancer products. We have worked on diverse cancer products. So despite our short history of the 3.5 years, we have made a lot of progress and achievements, and we'll continue to work on that. So please continue to support us. And with that, thank you very much.
네, 대표님 발표 감사합니다.
Thank you very much.
혹시 우상욱 대표님께
If we have any
궁금한
questions from the floor to Mr. Yu, would you like to raise your hand? We'll provide you a microphone.
혹시 질문 있으신가요?
Do you have any questions?
네, 없으시면 간단히 제가 그 짧은 질문 하나 드리면.
Then let me ask you a question. When explaining about the PROTAC,
가지고 있는 그 플랫폼들이 어떻게 보면
so the platform that you have, I think
적용을 할 수 있을 것 같은데 어떠한
in order to apply to PROTAC, they must be fused. So for the algorithm and technology that you apply for PROTAC, can you please briefly mention that? So as I said earlier, PROTAC consists of the recruit part, linker part, and warhead part, and these three should be combined well. to have a successful protect. And each of them could be generated with the AI solution. And we are highly interested in the different structures of the scaffold. And as for the linker design, based on the existing database, we try to find out the format that ensure the best flexibility. And so we make some prediction, and based on that, we do the design. And also E3 ligation and target protein, only with these two, you cannot really produce a good protect. And therefore, in order to have a good protect, we have to have AI plus physics base, and we have the entire structure to produce a good protect. So we work on understanding entire structure. And with that, thank you very much.
Moving on, I'd like to now invite the next speaker, Ms. Yoon So-jung, CEO of Standime, will give us to talk about to discover first-in-class drug candidates using workflow AI. Good morning, everyone. My name is Yoon So-jung from Standime. It is my pleasure and honor to be able to introduce my technology. I am given 20 minutes only, so I'm going to be brief and fast. My apology first before starting. I have not been in the offline meetings for such a long time, so I get more nervous than normal nowadays. I'd like to sit down when I make my presentation, if you allow me to. Thank you very much.
예, 양해 죄송합니다.
Thank you for your kind understanding.
오늘 발표드릴 내용은 다음과 같습니다.
So this is my topic today. So I'd like to give you the company overview of Standime. Then I'll be talking about the AI companies in the world, how they're working in the market. Out of them, the main technologies will be discussed briefly. Standime was first launched in 2015. Until today, we have finished getting the investment up to Series C. Thanks to the support by many investors, we have been growing so far. So based off of their support, we are now able to induce many talents to our company. So with those experts in the R&D, we are working so hard on our research. So if you see this diagram, you can see how we are consisting of, how many people are here, what kinds of people are here. Although we are just drug discovery person, we do have AI engineers as well as biochemistry experts as well. So we have a full gamut of the expertise here. The reason is because that although we are the AI platform company, I believe that we should be focused on the creation of the drug substances. So the AI-based substance making is our core essence of company. So we should be the best one in order to do that, and also we should have some knowledge about the papers as well, but also we should have the full implementation of the systems to the bio-pharma companies as well as other conglomerates. So we should be more attractive to those clients rather than to the academic field as well. So we are doing our utmost in getting the expertise. We are based in Seoul, city of Korea, but we are entering overseas markets in the UK as well as in the US from last year. So AI is more familiar to you probably as a keyword, and starting from 2018, we were fully engaged in the drug discovery as well. Many other companies are joining this trend, and R&D activities are quite heated nowadays. If you see the diagram here, so you can see the pharma research is announcing this graph to show you how the market is consisting of. So as of 2020, we have the top 5 companies or top 15 companies which are located in this graph here. So in the x-axis, we have the left corner to the left side. They have the companies of having the one step further in the AI for the drug discovery and the clinical development. And also to the right side of the x-axis, they have the end-to-end companies in such a field so that they can produce some final drug product. The y-axis is about the level of the output and where they are in terms of the focused areas. From the perspective of the pharmaceutical companies, at the bottom of the y-axis, getting closer to the zero, they are early discovery stage for the companies. They are just getting the output on that stage. To the higher portion, they are going into the clinical phase stages. So their outcome nowadays is actually seen in their clinical trials. So as I said that, what I am going to talk about is the first-in-class development for the discovery by using AI. What kind of efforts have been made by those companies? That's my topic today. So out of that, I can focus on particular technologies, but I can also give you the end-to-end efforts to make the discovery. I can give you the whole process of doing that. In this context, I can give you that we have our activities done by Standime. So if you see this, the workflow AI is the major platform that we have in our company. So this Standime is in the first stage of the systems. Considering the current technologies, Standime believes that the AI is the most optimal to have the best outcome in this area. Of course, sooner or later, we can also cover development down the road, but for now, we can have the discovery phase, which is the early stage. So here, we have to make our output as soon as possible to have the output of the high level of the substances. So in order to do that, we can have the end-to-end linkage of the processes, because one expertise only will bring us the results. We should have an integrated system, so we should have the standardized sequences of working here. So we believe that this workflow is quite important. So we have our proprietary system called MAGIC. This is a standardized workflow system. So if you see the bottom of the screen here, we should start with the targeting of the drug development first. To what patients, to what diseases or conditions. So this workflow will start from the identifying targets first. So the ask is the AI system to do that. So here we have the Standime best to have the design of the novel compound, and then we do the synthesis. Then after having the synthesis, the outcome of this is to have the trials of the various systems. Then we can have the first-in-class compounds. These compounds are actually early stage, as I said, because our outcome result is to beat the seeds of the next stage someday later down the road. So we can have the full groundwork for making the seed for the future development. These seeds should be commercially available as well. So in order to make that happen, rather than having these substances or compounds, we should have the identification what kind of substances are for what diseases or what disease classes. That will be the game changer, as I believe. So we have to know the target identifiable list that nobody knows yet. That will be the great starting point for our business. So we run down on the workflow. So our systems are quite parallel, so we can have multiple projects at the same time together. So you wonder why? Why is that we have to be this aggressively parallel in terms of the number of projects at a time? The reason is because the technology level we do have now should be working on getting the early stage work on the targeting of the compounds that we can work on later on. So even if these compounds have not been known to other companies or in public, we can get the ownership of this as early as possible, and then we get the targeting. Then we can go to the next stage, for example, having the collaboration or out-licensing. That can be our core capability, as I believed. So not just for the core thing, but also we can have the end-to-end services, and that is also the maximization of the AI capabilities. The biggest benefit of using AI is that any failure can lead to the next opportunity. So one failure will not be the end of the story. So we might have some kind of breakthrough problems, and sometimes we can go to the errors because of minor problems. But if these errors can be a source of finding new solutions, that's not the end of the story. So any failures can be accepted and used for the next round. But the reality is not so much favorable to that kind of failures so far. But I believe that the failures can be the textbook for the next stage trials. So what I believe here is that we can be intelligent in getting use of the lessons learned from the mistakes, and we can fail, and we can fail very well. So the first trial might be a failure, yielding nothing, but the second trial will give us the 50% outcome, and the next one will have higher percentage of the outcome. Then we will reach the point where we want to be. So as I showed you before, that is why we want to have multiple number of projects on a parallel basis. So having said that, what is the most important in this program? Of course, target discovery is really important. So the substance or compound identification is quite important, and it is being done by many other collaborators and also the partners. But that is also a red ocean, I believe, and we have some capability in that sense as well. Earlier than that, the ways to improve the quality of the substance or compound is to have the system to identify unknown substances or unknown compounds. That is why we are having the system of AI for doing that. So that is why we have stand-time ask system. So let me give you an explanation about the stand-time ask. AI is working on the identification of compounds. If the story ends there, you will not be interested. But we have one step further. We ask why it is identifiable. So if we are able to explain why these substances are identifiable, that will be the better success. So between AI and why, we find some methodology that can be contradictory in a sense. We have the gap reducing in many steps. So it's not the outcome AI as a black box coming out of. It is a white box of AI that anybody can look into transparently. That is the stand-time ask. We have two modules in a major way. So in terms of the data, what kind of substances or what kind of materials have been observed by the AI, they can have the different outcome. There are two things. First of all is the literature-based learning. They can get the results out of that. The second one is the low-level data or omics data. The clumps of the numbers can be studied by the system so that they can present new perspectives that no humans can see. So using these two modules at a time, we can get a new module and we can get the results. And that is actually the path followed by many other companies. But as I said, what is more important is how to interpret it. So we have our methodology that is unique to our company is to have the introduction of graph. So as you see here, so if we have the knowledge, we have to have a graphic explanation of what kind of knowledge is that. If we have the omics, we have the protein network, and we can also have the metabolic network, and also we can have the intelligent network. So they are explainable at the next level. So that's what we want. So inside the path level, that is actually the network of the knowledge. So if you get the idea from here, knowledge, it's something that you can learn from reading the papers. You can get the keywords and you can develop these targets. And that's the process, of the knowledge graph. So if you have the graph of the knowledge in terms of biology, you can identify what you believe as the most important ones as the key nodes, and they can become the nodes. And then you can have the interactions as edge. So between these core and edge, you can have the network building. Now you're based on the mathematical calculation, and you have the node and feature that are the basis of learning. So I'm sorry, and I feel dizzy a little bit. I can sit down if you allow allow me to. Thank you very much. So in the node and the pathway, you have the pathway feature that you can explain things from, so that you have the feature. So as you see here, network, we have the red dots here. Please look at the red dots. Red dots are the diseases, and the blue dots are the outputs. So under the current knowledge system, you don't have the lines. There's no direct edge between the 2 points. However, if you are studying from these modules, you can identify hidden edges. So these hidden edges can be seen after you learn these things. So it's not about the high score of the edges that can be visible. It can be based on the various pathways. Out of the pathways, you can see which is the best for explanation. So you can explain why and how you were able to get the network, and it is based on the contribution. So it shows how you can explain things based off of that. So we have the demonstration version, IFI cleaning, and we have the web page. If you come to visit our web page, you can have the trials hands-on for these systems. So this is a knowledge level graphic system. The next challenge we have to overcome is to have the fast update of the systems. We have to be speedy in capturing this information. In order to do that, we have to be based on the NLP, natural language process, so that we can have the very fast supply of the data and information. Based on the NLP, when the text is given, inside that we have to know the target, we have to know the disease, and we have to be able to map the relationship between the two. Then also using the pathfinder, which is a huge graphic, we have to put in there the data. So that's kind of module is needed. So here it's not simply about the target and disease and the relationship. Even if they have the higher frequency of the intermingled, that doesn't mean that they are eligible for this core system. Rather, you have to be based on the real relationship, the level of the relationship between the target and the outcome. So the information that you believed in the not relevant can be now relevant here. So you have to use the system in order to have the more relevant ideas. Now I talked about the targets, and so many of the researchers can read the papers, as I said, from others, and they can mimic the data given from these other papers. But here, what we have to propose is to have the further absorption of the knowledge after reading this, the next step. What kind of outcome we can elicit. So we knew that we have to use some kind of nodes. That is why we have the omics data. If we have the modules of the omics data, probably explainability can be taken out. However, here the graphic element, which is called PPI, can be used here, and also metabolics can be added here, so that we can increase the explainability. Here, as you see from the graph here, so we have the general networks and other knowledge networks. These general networks, however, are not reflecting the particular context that we're looking for. So we have to find the particular networks here. So on top of the general network, we have to have the major key players that have been known so far as the proteins. We have to mark these proteins. So on top of these marked proteins, we can add the omics. Then we can have the weight of the edges. But these edge weights must have a lot of noises. If you do not take the noises out, you can be confused with that. So you have to have the random walk information diffusion systems in order to reduce the noise, and you can be closer to the context. For example, gene expression level values can be taken out. With the noises taken off, then the ultimate output can be produced from here. So if you have a second look at this after doing that, it looks like general PPI network, but it can be based on the particular sub-module, or particular nodes or particular edges can be conspicuous more than others. So based off of that, in the new context, you can see that these targets must be important. This must have some kind of biological relationship, and you can interpret and explain things further out of the relationship. The last thing is out of the metabolic, you have the mixed systems. So we want to look at the metabolics here. In the genomic scale, we have the metabolic network is here. So this is coming from the general human beings to be constructed. We have such a model to do that. But as I said, what we want to do is the context-dependent ones. In order to reflect the context, we have to have each disease-specific omics data paired with the systems so that we can have this disease-specific results. So now we can do the simulation that we have done conventionally. So if we just stopped doing doing the simulation, and we will not be differentiated by others. But we can see that this kind of the systems see the only fluctuations of the systems that can be seen as major parts. So that's what we have done so far. What differentiates ourselves is to have the genomics system. So we have the flux in the genomic and the metabolic system are added using the metabolic information, and the phenon is added on top of our information. So if we have the addition of the data and SNP, SNP, then we can know that this is the gene for functioning some parts here. So we can identify what kind of functions according to the genes. So as I said earlier, so in summary, what I do is to give the target module in a big picture. Then also we also have the specific data based on the omics as well. So specific context can be reflected on this general map. Then we can identify the targets and the specific areas using stand-time-ask. So I think I'm punctual. I'm almost finished. So briefly speaking, we are doing our research and development in an ongoing matter, and these projects cannot be disclosed to you for now, but we do have a project that is being done as a co-pilot with a global genome company that I cannot disclose the name of. And we have the target of finishing it within the time period in our whole project. So these target genome that we developed are under the stand-time ask and stand-time best. And I've been talking about the target identification today. Thank you very much for listening. I am very sorry to sit down all the way. Thank you very much for understanding. So are you getting better? Less dizzy? Yes. Yeah, I know that everyone feels nervous after 3 years of absence of meeting in person. So if you have any questions from the floor to Dr. Yu, we will give you the microphone to your side. So any questions from the floor? To Ms. Yoon. Otherwise, I have a very simple question to you. Today, you talked about the stand-time ask only. Yes. Well, theoretically speaking, all the indications can be available in this platform, as I believe. But in the real world, you can have the limitations of the data available now. So do you have any specific targets in mind that you want to focus on first? And what kind of activities are you doing more actively and in indications and the target areas that you want to work with your stand-time ask? Yes, as you said, we think that the distribution of the data for learning is quite important, and we have to have various types of data available. That is the cancer area, so we can have ample data for cancer treatment, and fibro disease can be one of them, and we have two more diseases that we are focusing on. But as I said, we are not confined to indications in our system, so we can expand our systems, applications, and we can also work with many other partners to have the betterment of the in-house data. So we can have the curation of the in-house data further by working with other partners, and we are in the process of expanding such in the library. So currently, as you said, we are focused on the cancer and fibro disease. Thank you very much. Thank you for your excellent presentation and the answers. Thank you. Ms. Yoon.
Moving on, we would like to invite the Park Sung-soo, CEO of Dearden, for his presentation titled Sequence-Based AI-First Pharmaceutical Company Bypassing the Need for 3D Structure. So, good afternoon. I am CTO and Vice President of Dearden. My name is Park Sung-soo. Thank you very much for inviting me to this auspicious occasion despite bad weather. Thank you for taking part in the event. Dearden, we are also a AI-based solution provider for early-phase drug discovery. Our co-founders have great expertise in bioindustries as well as IT. So basically, we are founded by AI engineers. So we do have our unique differentiated approaches. And we are working hard to develop AI-based drug discovery solution. I'll briefly introduce to you my company, and then I'm going to talk about what kind of approaches we do have based on AI. And last but not least, I would like to share with you my learning points, some of the lessons and pains that I experienced. This is our company highlight. So we secured the 17 million Series B funding, and currently we have about 60 full-time employees. We have 3 AI integrated platforms. So target discovery, hit discovery, and the lead optimization. So we do have 3 optimization platforms. And early this year, we established our own R&D center in Dongtan area, so for synthesis. And currently, we have the 20 ongoing target studies with the 10 partnerships for co-development programs. We also have 4 in-house pipelines for early-stage drug design and development. As you know, AI has emerged as a strong player for drug discovery. This is because traditionally, when it comes to the drug development, time cost curve was very inefficient, that it was very difficult to achieve ROI. And one of the solutions in overcoming the traditional barrier could be AI. So we also, with the same purpose, in order to accelerate time cost curve, so to minimize investment and facilitate the time for achieving a break-even point and return. So this is what we are focusing on. And here you can see the market attractiveness. So as of last year, the market is worth to be about 1 trillion won, and it has grown by 30% every year. So by 2030, it will be grown to be 10 trillion won market. So with this vision, we also established our company back in 2016. And prior to 2020, we focused on technology development. But then since 2020, we have pursued the various partnership opportunities for co-development and received some investment as well. And last year, we established the Dearden USA in order to have a global presence and also to secure partnership with the global big pharma companies. And similar to the standard, out of 60 employees, half of them are AI experts, and the other half is for actual synthesis and experiment, and also the target discoveries for bioproducts. So we do have very balanced team. So with the employees with diverse backgrounds. So although we are the AI solution provider, if you only have the AI talents, then it's not really balanced. So we try to have the balance of the AI and the pharmaceutical experts. And we are also mentioned by TPI report. And this slide shows the companies that we want to refer, take as reference. So Dearden, here you see that we are one of the companies that other companies are trying to refer, a benchmark case. And currently, we are strong in preclinical pipeline, and we will expand our capabilities to cover clinical pipeline as well. And not just the Diogen, I hope that the other companies will also produce the tangible outcomes so that they can be listed as early as possible. Now, I would like to explain you how we discover drugs based on AI. So first of all, we do have a sequence-based drug discovery. We didn't pioneer, I wouldn't say that, but I believe we are the leader in this field. And traditionally, so we focused on the structure-based drug discovery or ligand-based drug discovery. But if you do not have the structures or no substances, then how can you discover drugs? So this is where we can rely on sequence-based drug discovery. So human protein, let's say it's 100, then what's available for drug discovery with known 3D structure, they represent only 11%. And the other 89% is protein with unknown structure. And here, what unknown means that it's 3D structures. Although you want to identify that, but it's difficult and impossible, or there are no needs for identifying the structure, or it's just too costly to identify and understanding the structures of the proteins. And even with the known 3D structures, 3D structure is just the beginning of the drug discovery process, and actually has to have a lot of diverse information in order to develop the actual protein. So, which means that the number of proteins available for drug discovery is very, very limited. So even without 3D structure, we try to create a model to predict the drug-target interaction. And this is what we call MDDTI, or molecular transformer drug-target interaction. And in the AI field, so especially with the natural language processing, transformer technology is widely utilized. And for the first time in the world, we applied this technology to drug discovery process. So here you see the free schematic drawing. So this is the 3D molecule structure. And if you convert that into the sequence with the smart format conversion program, then here you can see this kind of elements and atoms. However, with the traditional AI technology, it's very difficult to identify these sequence patterns because the convolution neural net with the CNN, if you learn this sequence, then although the sequence is near and close, but the flow ring, FNCL, are apart from the sequence, but structurally they are very close, or the vice versa. So there could be some gap. So in order to learn the structure, we actually have to train the algorithm with the global structure. So we use the CNN to overcome this barrier. So this is our sequence-based approach, and I would like to talk about some case studies. I believe that one of the incidents that enhanced our reputation is that after the outbreak of the COVID-19 in USI in early 2020, Diogen, at the time, in fact, the COVID-19 didn't really have the known structure. So in January, there was no 3D structure available for COVID-19. And in order to identify the target materials, candidates for therapeutics, only with the COVID-19 sequencing, we were trying to develop the substances that could respond well to the COVID-19. So we did the screening, and we came up with the list, and we published the list. And this is one of the events that enhanced our reputation. And one of our substances that we identified were one of the top 5. And this paper, we actually wrote that in 2 days. So 1 day for the experimentation and second day for writing. and it was cited for more than 600 times, and it was selected as a top cited paper in the journal that we published our paper. So we input the amino acid sequence with known 3D structure, and then we run deep learning algorithm. And we then evaluated the affinity score, activity score, so whether it works as an inhibitor or activator. And so this is the output that we can produce. And the AI fundamental technologies that enable this solution includes the molecular transformer, drug target, active training. So what kind of pair of the drug targets have to be learned? This can be identified through the active learning. And also the reliability of the results have to be measured as well. And also the drug target interaction. And also the location of interaction is very important. And therefore, we use the cross-attention technologies for drug target interaction.
저희가 세 가지 플랫폼을 가지고 있습니다. 디어트랜스라는
So we have 3 platforms, and DearTrans is for the target discovery, and the input includes the genome data. And once you input genome data, then we identify what kind of markers could be the significant biomarkers through the genome analysis. So we created the AI model called XX, and using this, we can do the MOA analysis and drug discovery and target discovery. And DRGTI, this is again sequence-based drug target prediction platform. And so you look at the protein sequence data and also drug sequence data. So once we input these 2 data, then it identifies the 3 different kinds of interactions. And for this, we can have the nuclear discovery and drug proposal, drug repurposing. And this is the MOIQ, and this is called this stands for more equalizer, and this is for drug development. So I just talked about the DTI. So even if it binds to the target site, not every substance can be drugs. So the toxicity as well as the ADME, these different properties have to be optimized and screened in order to identify the good candidate for molecule drugs. So we do have a technology for that. So DearTrans, DRGTI, and MOLE-EQ platforms. And we also have secured a lot of patents in relation with these 3 platforms. And currently, we have established the WebNet. The reason why is because in order to turn the failure into success, in order to create a virtuous circle, we thought that we should be involved in the target discovery, hit discovery, and lead optimization, as well as the wet lab. So if we could do the synthesis in the actual wet lab, so that the results of the wet lab could be processed back and feed back into the target discovery process, so we can create a virtuous cycle. And also all the data that are produced from the wet lab, they are also feed in to our database, in-house database, for the betterment. And now I'd like to share with you the 2 case studies. The first one is about the dementia. So this is to identify the novel protein with known 3D structure in relation with the dementia. And for this novel target protein, using DRGTI, we found sheath. So we did the heat discovery prediction for about 2 weeks, and then we came up with a list of the candidate materials. And then we did the experimentation with the 10 to 18 compounds, and 2 of them produced the heat. So we could not disclose the structure of the substances here. So we did the 46 nanomole prediction, And in fact, the actual value was 60 nanomole.
아밀로이드 베타
And
감소는 피코몰 단위에서 보였습니다.
amyloid beta reduction was also found at picomole
또
level.
다른 케이스는 저희 프로탁을
And
했던
the second case is
이제 뭐 여러
with
가지 뭐
PROTAC.
스몰 모레큘에 집중을 하고
So
있지만
although we
스몰
focus
모레큘
on
안에서도
small
여러 가지
molecules,
모달리티가
but
있기
within
때문에
small
중에
molecules there could be diverse modalities, and one of them is the PROTAC. So we did the case study with PROTAC. So with the PROTAC, we predicted the binder, and with the existing SRI bounders and linkers, we did the experimentation on target protein, whether the integration is smooth, and we demonstrated and validated that through the experimentation.
그래서 이제 그 4분 정도 남았는데요. 그래서 제가 했던
I have 4 minutes to go.
느꼈던 거를 이제 좀
And now, so I'd like to share with you the experiences that I got throughout this process, or lessons learned.
뭐이 자리도 그렇지만 이제 AI 드럭 디스커버리를
So as for the AI-driven drug discovery, we have the history of 4 to 5 years. And in fact, for the past 4 to 5 years, there were a lot of booms and new startups being built pursuing AI-driven drug discoveries. And if you look at the number of startups specialized in AI-driven drug discovery, you can see that from 2019, well, from 2018, there was a slight decrease. And year 2020, we only have very few startups being established. This is because of the fierce competition. But anyway, the number of the startups being built is declining, and also the amount of funding and investment is also dwindling. It's because of the bad economy nowadays. And also for AI drug discovery field, it is no longer being expanded. Rather, I think now is the time for us to see some tangible outcomes. That's why the investment has been held back. Then what kind of approaches we have to have for the future? And for this, I would like to suggest the 2 models, general model and narrow model. So general model means that we have to target general disease, general target, and big data. And if you follow this approach, compared with the narrow model on the specific disease, specific target, and small data, there could be some performance gap. So here you can see the difference. So the accuracy is like 1.3 times higher with the narrow model than general model. And error ratio, again, declined by 40% more in narrow model than general model. And also for data, do you have to put in as much data as possible, or quality is more important than quantity? So we have to have a high quality data. So in the beginning, we thought that the amount of the data is very important, and therefore without modification and without quality enhancement, we try to put in as much data as possible. But nowadays, we have shifted to improve data quality. So we actually decrease data samples, and we try to produce high quality data. And with that data augmentation, we used the data, and we found that the accuracy actually went up by 1.2 times, and loss score decreased by 0.7 times, or 30%. And also, we have to think about whether we are targeting producing diverse drugs or real drugs or practical. And in terms of design, I think this is very important. So when we design the drugs, we have to design diverse drugs, different ranges of drugs, or real-like drugs. And I think that there are always trade-offs. In the beginning, we try to have very diverse drug designs, but nowadays we focus more on real-like drugs. So the drugs that are available for synthesis, available for accessibility. And depending on modalities and processes, I think that things could be different, that in some modalities and processes, the diversity is more important than reality. But our focus currently is on real-like drugs. And after we achieve that, we will go for diversity. And with that, I would like to conclude my presentation. Thank you.
네, 부사장님 감사합니다.
Thank you.
네, 혹시 박성수 부사장님께 궁금한 사항이 있으신
If you have any
분은 손을
questions
들어주시면 저희 스태프이 마이크를
to Mr. Park Sung-soo, if you raise your hand, we'll give you the microphone.
예, 세 분 연사분의 발표 전부 다 흥미롭고
I really enjoyed all three
AI와
presentations.
신약 개발에 관심이 굉장히 많은데
I am highly interested in AI-based drug discovery. I'd like to ask a question to all the speakers.
지금 질문을 드리고 싶습니다. AI를 이용해서
So using AI,
신성이 높은 아웃풋을
reliable, in order to produce a
트레이닝에
reliable
들어가는
output,
인풋의
input data
양과
for
질이 중요하다고
training, the
박성수
quantity and
부사장께서
quality, I think is very
언급을
important.
해주셨는데요. 아까 말씀해주신 내용을 보면은 그 이전 개발
And
사례 거기서 나온
if you look at the
물질들의
previous
뭐
development
구조나 그
cases,
물질들의 어떤
you know,
효력
like the structure, the
안정성,
efficacy,
그리고 실제
safety
임상
of the
결과까지도
substances,
인풋으로
and
사용할
even
수가
the
있고,
clinical trial
그리고
results can
리터레이처
all be
결과가
used as input data,
발표된 연구 논문들에서
and also
나온
literature-based
결과들이고,
data
오믹스
and
데이터베이스들을
omics
기반으로
database
트레이닝을 한다고
can also
하셨는데
be used as an input data
실제로
for
탐나치
training.
논문에서 이제 발표되는
And
결과들의
results from the
절반
top-notch
미만이
papers,
재현 가능하다고 알려져 있고,
only half of
다시
them
말하면은
are
절반 이상은
available for
거짓말이다라고
formulation. In other
이야기할
words, the
수가
other
있고요.
half is
오믹스
only
결과들도
hype, or
저희가 이제
let's say
살펴보다
lie.
보면은
So, and even
대부분의
for the omics
오믹스 결과에서
database,
어떤
most of the
실험적인
omics data,
개발하고자 하는 질병의
the
모델이나
experiment
세포
conditions
혹은
are pretty
종과
apart from
잘 맞닿지
the
않는 경우도
actual
많습니다.
disease that they target
그리고
or the
이제
species that they
이전
target.
개발 사례들을
And also
가지고
if you look at the-
트레이닝을 하고자 하면 대부분
and
참고할 수 있는 데이터들은
Also, if you
공개된
try to
데이터에
train
한정된
the
경우가
machine
많습니다.
with
공개된
the
데이터와 그것은
existing
주로
researches,
어느 정도
then
가능성이
you have to
있다고
rely
생각이
on
되는
disclosed open
선도 물질 혹은 후보 물질에
data.
대한 개발 사례이지
So these are the
그 선도
development
물질 혹은 후보
cases
물질이
for
도출되기까지
the
이제
candidates
뒤에
with some
적게는
viability.
수백 가지 혹은
But
많게는 수천
there
가지 이상의
are
물질들에 대한 데이터는
thousands
공개가 되지
of
않기 때문에 이제
substances
활용하기가
where
어려운
data
것이
is not
현실이라고
disclosed, and
생각이
these
듭니다.
data
이런
are very
부분들을
hard to be
세
utilized
분께서는
for
어떻게 타개를 할 수 있다고
training and drug
생각하시는지
discovery. So
질문을
how
드리고
can we
싶습니다.
overcome these barriers?
네, 제가 먼저 답변 드리겠습니다.
I will answer that.
두 가지 저는
I like to.
했는데 그 오믹스
I believe that there are two
데이터의
questions, in fact.
퀄리티 문제.
So the
그다음에
first one is about the
데이터
quality issue of
예,
omics
어떤
data, and then
생성되는 데이터
you also
대비해서
asked
필요한 데이터가
about
많다. 그렇게 두
the
가지
data available for utilization
첫 번째-
versus the
첫
data
번째
that are
질문은
produced.
사실 저희는 오믹
So for the first part of the question, which is about the quality of the omics data,
그 퀄리티를 높이는 작업을 할 수는 없거든요.
so AI cannot
사실은
enhance the
퀄리티
data
높이는 작업들은,
quality.
또 다른
Data
또
quality
분량들이
enhancement should be
당연히
done
들어가서는 해야 한다고
in
보고요.
other fields.
두 번째 질문의 답변은
And
당연히
for the
그렇기
second
때문에
part,
실패한 그 하나의 약물을 계기 위해서
let's say that the one
실패한 약물들을
substance
피드시키기
fails
위해서는 그 제약사 쪽에서
to be
당연히
developed as
공개해야
a
되는데
medicine,
공개하지
and in
않죠.
order to
그래서
feed in this data,
저희가
the pharmaceutical
여러 가지
companies
방향으로
have to
풀고 있는데
disclose
그게
such data, but they are
제품에
reluctant to do
협의체
so.
같은 걸 구성해서 풀
So
수도
maybe
있고,
we
근데
can
저희는
establish a
두
council
가지
to
접근을
persuade the
세 가지
pharmaceutical
접근을 하고
companies
있습니다.
to
첫
disclose
번째는
their failed
인하우스
data,
나온 데이터 쓰는 거.
or you can have an
콜라보레이션에서
in-house
나온 데이터
database
쓰는 거. 그다음에 세
so that you can utilize
저희
all these
플랫폼을
data.
제약사에
Or
넣는 거죠.
let's say that we can
제약사의
provide our
데이터를
platform to
저희가 받는 게
pharmaceutical
아니라
companies.
저희
So rather than
플랫폼을
receiving
제약사에
data
넣어서
from the
그
pharmaceutical
제약사에서
companies, we
저희
can
플랫폼을
establish
가지고
our
데이터를 쓰게
platform
그렇게
in
하고 있습니다.
the pharmaceutical companies so that they use our platform and put their data in there.
네. 저는 아까 이제 질문 주신 것 중에
For
저희가
the
오늘 타겟
question,
발굴 관련해서 설명을 드렸기 때문에
I actually
아까
talked about the
오믹스나
target
이제
discovery
그런 논문
and
관점에서 좀 말씀을 드리고 싶습니다.
but in terms of the omics data, I think that your question is about when the data is garbage, and if the AI is trained with the garbage data, then can it really produce a reliable data? And also you mentioned that a lot of we have the problem with the data reliability, and also omics data does have a lot of noises, so many of them could be insignificant. I'm not here to defend AI technology, but from the external perspective, let's say, the paper data and omics data, the quality the fundamental issues in relation with the quality of the data, it is solely responsible from the AI? Maybe not. So there could be some biases and also the noises in data apart from AI technology. And so what I would like to mention here is that AI, yes, if it's trained with the garbage data, then it will only produce the garbage data. And therefore, the quantity and quality of data is important. But a lot of knowledge and data that we have produced so far throughout the human history, not all of them are garbage. So although we do have some garbage and noise, but still we have to make the best use of them. And making the best use of them means, let's say researchers, they work hard, they did a lot of extractions, and they created the collective intelligence. But I think that what they have done was still within the realm of human beings. But AI, it can actually provide a different output, even from a fragmented, obscure database. And AI can actually have some special function that human beings cannot. So the quantity and quality of data should be improved, of course. So in the beginning, our focus was to put in as much data as possible, but then our focus today is to curate the data so that we can have high quality data. And also the machine learning, they actually monitor the different variables all at once, and therefore they are not really dependent on one single data or one bias. And so compared with the human interpretation, I believe that they can cancel out all different noises more effectively and come up with the reliable data. So in the sense, I believe AI does have some upper hand in this field. I will also answer the question. So the two previous speakers, I completely agree with what they said. And I would like to share with you my own experiences here. So garbage in, garbage out, yes, this is applicable not just to AI, but it is also applicable to molecular dynamics. And I think that for people who are involved in the in silico, garbage in, garbage out, this is something that, let's say, it's our destiny. So I also have some suspicion whether all of our experiment data is really clean and complete. So in the sense, I'd like to share with you my experience. I was in the postdoctoral graduate course in the North Carolina, and I had an anticoagulation system, and I researched on that. And we have two professors, and both of the professors wrote the papers on more than 500 kinds of papers on thrombin generation, and their conclusions are different. So in order to understand that, what I did was that I read the experiment conditions of the two professors, but still I could not come up with a conclusion. So eventually I relied on mathematics. So at the time I didn't use the AI, but if I used AI at the time, then I think that I could rely on AI. And also back in 2009, when I arrived in the COAX, I heard about IBM Watson, and they said that the IBM Watson opens a new frontier, and I was in the attended seat. And at the time, what I felt was that, let's say that there is a specific cancer, and if there is a controversy existing between the two different schools of papers, then what kind of data should I use? You see, the IBM Watson, it actually uses a lot of polluted, contraindicated data, and that actually compromised the prediction accuracy. And therefore, the reason why we have carried out the researches on AI physics is because we are highly dependent on AI, but for the AI prediction results, if we could utilize the mathematics, physics, and chemistry, you can also validate the results produced out of AI. In that sense, I think AI and physics are in sort of the complementary relationship. That's why we focus both on AI and physics. And one more thing that I would like to mention, if you give me the time. So I talked about the AlphaFold, and we have used that for the structure prediction for the GPC, or even with the X-ray crystals. It is very difficult to predict which one is active and which one is inactive. So even AlphaFold structure prediction, depending on what is used as an input, the result could be very different. So your question is very critical, and a lot of people are working hard to overcome that issue. So aside from data, I believe that we have to have diverse validation solutions.
네, 혹시 답이
Thank you. And did they answer your question? Thank you.
이제 세 분한테 발표 잘하셨습니다.
Originally, we wanted to say compliments to the speakers and make arrangement for the next session, which is the panel discussion. But this naturally moves to the panel discussion that we just have finished. So time is almost up. So we think we have already finished our panel discussion so far. AI is not a concept that is really far away from our daily lives, because on television, on the commercial programs, we are watching artificial intelligence that is being viewed. And also the announcers and idol singers are also AI-based ones. From time to time, they are gathering their popularity. Having said that, the three presenters from three companies here are some of the very abundant AI-related companies in Korea. Actually, the bio is the most important and difficult one to tackle in terms of AI, because we are talking about people's lives, and that is most risky and most difficult items that we have to solve out of AI. With today's presentations as a momentum and starting point from the Diam, PharmCat, and Deergen, we now know that there are so many others who are working with the existing drug development companies, and their mission to make Korea a better country in terms of the biotechnology. We give our compliments as well as encouragement for these companies to do better and more. Thank you very much for your participation, and please give them a big round of applause.